Hi, this is Miss Litton, and this is my wonderful APBio Period 1 class. Say hi. Hi. And we are continuing on with Chapter 24. And Chapter 24 is about flowering plants, structure, and organization. And in our previous discussion, we went over all of these different tissue types, so let's just review it really quickly. So meristematic tissues are tissues that are capable of cell division and growth. And permanent tissues, we looked at 3 over here. We looked at parenchyma, collenchyma, and sclerenchyma. And parenchyma was the general plant cell that if you put a plant in a blender and pulled one cell out, chances are you were going to pick up a piece of parenchyma. And I gave you an example here with an apple. It could be used for storage. It could be photosynthetic. Collenchyma, this would be something you would find in celery. They have secondarily thickened walls, and the walls are actually the green that you see in this photograph. The cytoplasm is the darker color, so that shows you that it's thicker, it's more for support. And then sclerenchyma, these are dead at maturity. Definitely for support and structure, enclosing a seed, but yet you can also see them in pairs. Then on our xylem and phloem, could the youngest bio buddy please differentiate between xylem and phloem? Go, youngest bio buddy. Okay, and I can't remember if we went over this in the notes or not, but which one has to be alive to work? Do you know which one? Phloem. Yeah, phloem has to be alive to work, and xylem works just fine dead, no problem. And we're going to discuss transport of water, or xylem sap and phloem sap. Xylem is transporting water from the roots all the way to the tops of the leaves and the shoots. Good. And we're going to talk about that, the TATC theory, in chapter 25. And so we'll talk more about, why it works when it's dead. But phloem has to be alive to work. And when it moves sugar, it's a process of translocation. And it works a lot with the osmotic potential of the cell. And we'll discuss that also in chapter 25. Okay, so phloem needs to be alive to function. We looked quickly at a few pictures. This is showing you xylem. You could have tracheids, which are smaller with tapered ends. And then vessels are like PVC pipe that you stick together. And phloem, you can see there's a sieve tube, which is this portion right here. And this is where sugars are actually transported, but it is controlled by its cell that's connected to it called a companion cell. And again, we'll hit more of that in chapter 25. Okay, and this is easy, but we'll just get started. Okay, please let this be 100. Is it going to be? Oh, check your bio buddy's clicker right now. If they don't show, start ringing the shame bell. Oh, I think I need to erase that one. I really do. Good job. You mean you don't like me, you guys? That's me. Yes, I'm a natural blonde. All right. So, just get an overview of plant parts. Why don't we have the oldest bio buddy take care of everything above ground, the shoot system, and the youngest bio buddy, you take care of the root system. Go ahead. And you have a picture in your notes too.
Primary is just the one that goes straight down.
All right. So if you look, this is showing you, in this picture you can see clearly that as much as you have below ground is usually how much you have above ground. The roots and shoots are usually about equal. Here on this shoot, can you see how the leaf is coming right off the stem? Can you see how it's just directly coming off? That is typical of what you would call a monocot. Whereas in this previous picture, do you see how we had a petiole that attached the leaf? That is typical of old school name dicot, modern name is eudicot. Okay? And monocot and eudicot, or the old school name dicot, come from cotyledons, and these are seed leaves. Now, what does the seed enclose? The embryo. Okay? So, now, what do leaves do for a plant? Catch light. What's that process called? Photosynthesis. And by doing photosynthesis, what are they providing for the plant? Energy, sugars, right? But when you're in a seed, can you do photosynthesis? No. So you need a source of nutrients, yes? And when the seed is first formed, there's something called endosperm, which is going to provide nutrients for it, but it moves into the cotyledons, either a single one, which is called a monocot, or two seed leaves, which is called a dicot or a eudicot. When you look at a peanut, you know how a peanut breaks in half? Because when you eat a peanut, you're eating peanut babies, right? Okay. So, you know how the peanut breaks into two halves, or a walnut breaks into two halves? Those are dicots. There's two halves to the seed. Or a bean, when you bite on it, like a lima bean, you bite it, it bites in half. That's a dicot. Okay? Monocots are when you have just one, it's solid in the center. Okay? And monocots would be, are sunflowers monocots? I think, yeah. So those are when it's just a solid in the center, the food source. But here's the thing, there are other characteristics that go along with that. And dicots or eudicots, I'm going to use those interchangeably, they have petioles like this, whereas monocots don't. Like corn is a monocot, it comes right directly off the stem. I also want you to look at these leaves. This is called, you don't need to memorize this, pinnate venation. Can you see right in here the veins right here? Okay, it could be pinnate venation, it could be palmate venation like this that you would see in a eudicot, but in monocots, all their veins run parallel. So think about like a blade of grass is a monocot, how all the veins run parallel. So you could tell the difference between a monocot and a eudicot just by looking at its leaves. But the name is coming from the embryo. And one thing, and I have more to show you on this in just a minute, but you can see right here, one of those things is the petiole, but I wanted you to see this. Can you see the venation a little bit in these leaves? Parallel venation, that's a monocot. When you start walking around on campus, I want you to identify to your friends, clearly that is a dicot, okay? So, and you can give them evidence, claim evidence, reason, okay? Use your science when you're out there, okay? So, is this a monocot or a dicot? Dicot, what's another name for it? Good, and how do you know that? What's your claim, what's your evidence for this claim? Yeah, I can see the veins are out here. Okay? If this were a monocot, everything would be parallel venation. Okay? All right. So look at here. Angiosperms are flowering plants. They are divided into two groups. You don't have to text me what they are, please. Okay. I owe you something though. Go up to standard tissue, organs of flowering plants. Vegetative organs you have root, stem, leaf, perform functions that allow a plant to live and grow. Flowers and fruit are reproductive organs, Okay, and then here are what they're divided into, monocots and eudicots. And you can look at root, stem, leaf, flower. You can actually look at the seed itself and how many openings there are. And I would like blue to be the monocot, and I would like slate to be the eudicot, and role play. ping pong back and forth for these different structures. Go ahead, quickly.
Okay, so unicorns have a split seed, monocots will symbol like unicorns have a between organ and xylem. Xylem and phloem are interrelated. And then for unicorns, it's like in a ring, and then for monocots, it's like all over the place. Unicorns, there's things that like have like not parallel, so it's like all over the place. And then monocots, it's unicorns, and then unicorns, there's monocots, it's 5, and then monocots, it's 3. So 4 is, and then there's 3 schools of 4, there's 5.
All right. When you hear vascular bundles, what are the vascular tissues of a plant? Xylem and phloem. Good. Xylem and phloem. Didn't happen inside. Okay. So when you look at things here, you can see the vascular bundles are scattered in this situation in the stem. They actually look like little aliens to me. Do you see one right here? It's on close-up. Okay. That's a typical monocot stem, whereas a eudicot or dicot stem, their vascular bundles are in a ring. And let me tell you why. That ring has vascular cambium in it, and it can generate more xylem to the inside and more phloem to the outside to allow for secondary growth. Monocots typically only grow one season, and then they're it. Sunflowers grow that one season, and they're done. They grow straight up because monocots do not have an organized vascular cambium that allows for secondary growth, whereas dicots do, and they can branch off in different ways because they still have meristematic tissue in a ring in that area. And it's in a ring in this area. Now, there's an exception to a monocot that lasts a really long time, and that is a palm tree. But think of how the palm tree grows. Straight up, right? Straight up on a palm tree and up on the top. Okay? It doesn't branch because only dicots / eudicots can branch like that. Blue, tell them 2 things you've learned. Anything so far?
Just put it on something next to it.
So, just, yes. What? Clover, like 4-leaf clover? 3-leaf. I think those are eudicots. I don't think they have parallel venation. Google it. Look at the leaf. You'll know right away. You should be able to identify these things like, oh, totally. No problem. What are these, monocots or eudicots? Eudicots, okay? And this one has what's called pinnate venation, and this one has palmate venation. Remember, monocots are only going to have what kind of venation? Parallel. That's it. Okay? So let's play a little game. Put it in the basket. Yes?
Is it monocots that curve, or is it the eudicots that curve when they
Both can.
Oh, both can.
Yeah. Okay? So, you don't know this part. That's okay. What is this? Monocot or eudicot?
Eudicot.
It is. What do you think this one is?
Eudicot. Eudicot.
Eudicot?
Yep.
Okay. And what about this one?
Eudicot.
Okay, and watch what happens if I put it here. No. Okay, what about this one? See, what you don't know about this is these are roots, and you don't have to memorize root structure anymore. Hi, sweetheart. You don't have to memorize root structure anymore, but eudicots have stars in the middle of their roots that look like that. And you know what the star is? Xylem. Sure. This period?
Yeah.
Okay, got it. Check though, I already have 2. They might say no, so talk to Miss Randall about it. She probably will say, oh, for me, she'll do it. And it's you, so she'll do it. Okay, but check, make sure, because I don't want you to get kicked out. Okay, so what one is this one? Eudicot, and this is a monocot. What about this one?
Monocot.
And why do you say that? Scattered vascular bundles. And what is this one?
Eudicot.
I agree. What is this one?
Isn't it?
What do you think?
4, 5, 6.
Why?
6.
Multiples of 3. Let's see. It is. What's this? Yeah, it's corn. What's this one? Yeah, it's, you can't see it very well, but it's got Oh, okay, good. Whoa, I set it up wrong. What's going on?
It looked good.
Don't trigger me.
You did a really good job on that. I'm super proud of you. Way to be a cotyledon.
I'm putting a text box in. Okay.
What's today? 13. Friday the 13th. January, right? February, March, April, May. 4 months from now, you'll be done with your AP exam. You'll be like almost done with your final even. Yeah, you'll be done with your final 4 months from now. Countdown. I used to keep on my AP page a countdown to the AP exam, so every time you had to go on it, you saw how many hours. Let's go, Marlon. Ah, most of you got this right. C is the right answer. And monocots, their vascular bundles are not in rings. They are what? Scattered, right? Okay, so it's just to the reverse. Somebody said B. Leaf veins parallel. That is true for monocots, and they're net veins for eudicots. So B is true. E. Yeah, no. All right, check with your bio buddy. Do they get praise or gong of victory if they don't show? Give them the Shambo. You don't need this for the AP exam, not at all. But just ping pong back and forth. Slate, you go do the first and the last one, and Blue do the middle one, just to get your brain all in the same place. Go. Does that make sense to you? Okay. Think perennial or permanent or something along those lines. Okay. We're going to talk more about hormones and flowering in the process. We're going to talk more about that in our hormone chapter. So on your notes where it says plant growing seasons, controlled by 2 flower-inducing genes, leafy, I like that name, and apatella 1. When these genes are on, remember how we learned to control genes, right? Yes. When these genes are on, the vegetative growth stops and flowers are induced. And flowers are induced. Three, 2, 1. Good job, sclerenchyma. Parenchyma is your general plant cell, right? I know this sounds stupid, but this is how I always remembered it. Parenchyma, I can still call them because they're alive. Call parenchyma. Sclerenchyma is dead. Okay, that's how I remember that. I definitely would not pick D, but somebody did. Why wouldn't I pick D for sure? What do you hear in that? Meristem. Meristematic, these are tissues that are continue to grow. Okay, so definitely you would not pick that as non-living. Oh, dang it. really set this one up. Wow, okay. Yeah, I just went over this. Meristematic tissues are constantly dividing. A big group of you got that, but 5 of you did not. Check your bio buddy and be encouraging as you shamefully ring the bell. All right, now we got an overview of all the different plant tissues. Now it's just a little bit of detail on each one of those. There's a lot of miniature words in here where you purely for support. I will not quiz you over that. They will not test. It's just if it helps you in your background or understanding. So we're just going to real quickly look at roots, stems, and leaves. So starting with roots, the purpose of a root is to anchor, support, and absorb. Anchor, support, and absorb water and minerals. It also produces hormones and can store carbohydrates. Anchors, supports, absorbs water and minerals. It can produce hormones and it stores carbohydrates. And the size of the root, the same amount that's below ground is probably what? Or above ground is below ground. And when you look, you don't have to memorize roots anymore, but when you talk about mitosis, if we looked at a slide, typically they have you look at an onion root cell. Did we look at some cells in mitosis, like any of the ones on the screen? And they'll pull them right here from the zone of cell division. Then there's a zone, so cell division, what do you hear in that? Dividing. Zone of elongation. Getting bigger. And the reason I'm going over this with you is because you're going to need to understand a little bit of this in order to understand another chapter on hormones. So the process of elongation and the process of cell division, either one are going to make the plant be what? Bigger. Either more because you're dividing or you're taking the cells you have and making them bigger. Okay? And then the zone of maturation is exactly what it sounds like. It's a mature region. It's not moving as much in the soil. It's just specializing. And this is where you're seeing these little itty bitty root hairs. And this is an adaptation because root hairs increase the surface area, which makes it easier to what? Absorb. So that would be the significance. You know, it's not like you have to memorize these, but here in the zone of maturation, that's where you're going to see those root hairs. So adaptation, extensive root hairs to increase what? Surface area for water absorption. All right, again, you don't have to differentiate between monocots and eudicote roots anymore, but one thing that is the same that we're going to talk about in chapter 25 that links it is this part right here. It's called the endodermis. And this center part of a root is called a stele. In the center of the eudicote root, you see what kind of shape? A star or a cross in the middle, and that's the xylem in red. The blue is the phloem. Here you will see the xylem here, and then the phloem outside of it in the monocot. And this part right here is the endodermis. The endodermis is where you're going to helps with water regulation that we'll talk about in chapter 25, and this is why. Each cell has a layer of wax around its end walls, layer of wax. So water cannot go between cells anymore. They have to go through a cell. And that becomes very significant in water, right? You can just sit in my office, okay? And if you want to, you can close the door, so you don't have to listen to me. And then this part, cortex, is where nutrients would be stored. If you took a look at these pictures, would you be able to guess which one was a monocot or a eudicote? And Slate, you do that. Talk about these pictures.
used to, yeah. And then that's because they're like,
And what are the blue rings that I just made? I just talked to you about it. Endodermis. Okay, now you're going to remember this because a layer on the outside would be called the epidermis, and a layer on the inside would be called the endodermis. And it's there that you have waxy cuticle. And this relates also to your lab, is that water can go between cells and around and between cells like this, and in any one of these pictures. And again, this chapter 25, it's called the apoplast when it moves that way, when it moves around and between cells. Now, another way that it can move is actually go from one living cell to another living cell to another living cell, and it's inside, cell to cell to cell, the water is moving, and that's called the symplast. So apoplast, think like apple, and you're going around the apple, and symplast, you're going in, cell to cell. Which one do you think is more regulated? The symplast, because it's inside the cell. Here's the deal. Water can move both ways, apoplast and symplast, until it reaches the endodermis. And it can't use the what anymore? Apoplast, because there's a waxy layer, and it has to use the endoplast, symplast, I don't know what I'm talking about, the symplast, and then that can make it more regulated right there at the stele where the xylem is located. Good, good. You're learning things in the future.
How do you spell it?
Okay, and this is just showing you a close-up, and this would be right here, the endodermis, this right here. The layer of cells just inside here is called the pericycle, and that's where branching roots come from, but you don't need to know that. Okay, so you already understand this. Blue, go ahead and explain it.
That's the legacy you want to leave.
All right. You good? Yes.
For the first one, like the one on the top. So
This one?
Yeah, so it would start being like the apple or whatever, and then it would end up being the like one where it goes through, right? Like.
It can be either or, and they're just showing you one stream of water, but like water is everywhere. Some is in a cell, some's going around the cell. It can be everywhere. So you don't have to worry like what comes first or second, but the key thing is water is completely unregulated out in anything outside of the endodermis. It's unregulated. That would be really bad for cells like us because we don't have a cell wall to help protect us from variations in turgor pressure, right? They are more adapted to that. But at the point where it enters here and gets inside the stele, that central cord, then it's going to be regulated because it has to pass through this living cell barrier that's either allowing water in or not. Now, I just want to see, because this is, again, a chapter coming up, but I want to see if your brain follows. What could make, there are no water pumps, right? We talked about how do things get through a cell. They can go what? Through the phospholipid bilayer. It can use a channel or a carrier. Yeah, whole membrane, endo and exocytosis. There are no water pumps. There are things that allow water to move more readily. What do we call those? Aquaporins. But there's no pumping mechanism. So how do I motivate water to move? Water potential. Okay. So if I want water to come in here, what do I need to do? I could actively transport other items, and water will want to what? Follow. And that's exactly what happens. That's how you get water in roots. Water goes from a higher water potential out here to a lower water potential here to an even lower water potential here, and that's how you can get water to move into a root. And it can't go back out because of the endodermis, so the only place it can go is up. That generates what's called root pressure, and that's why if you cut a tree down, it bleeds. Because they're still drawing water in. Are you getting that? Okay. Yes?
How does it lower the water potential? Like, where does it get?
From the soil. Yeah. Yeah, that's where it will get things to pump in. Yeah, good. Super smarties. I'm so smart of you. Okay. You don't need to memorize all of those things. I'm going to keep going on that. So root adaptations, anchor, absorb, and store. There are all kinds of adaptations. This is called a taproot when there's just one main root. Obviously, this looks fibrous. There are prop roots, above ground. These are, if you Google this, you would get a better picture close up. But these are basically these trees, okay? And what happens is they live in real swampy areas. Their roots are growing down and back up. In order to acquire what do you think they need, those roots need? Not sunlight, they need what? Oxygen. For what process?
Cellular respiration.
Oh my gosh, they need oxygen for what? Cellular respiration. Okay, so sometimes you will see adaptations like that as well. All right, moving on. Oh, here's showing you some prop roots. Here, these are all roots coming up. They've gone down and back up in order to get oxygen. And here, these are symbiotic relationships, mutualistic relationships. It could be between a fungus and a plant root. They can increase the, here, we'll just put it in the notes. Root symbiotic relationships, mycorrhizae. Fungus provides water and minerals to the plant. And then you can also have the fungus, by having all of this mycorrhizae right here, they increase the surface area for absorption. What do you think the plant's paying back with? Because it's mutualistic. What could the plants give the fungus? Are fungi autotrophs or heterotrophs? Fungi are what? Heterotrophs. They're in the soil. What could the plant give them? Nutrients, sugars. So in trade. So the fungus will give them water and minerals by increasing their surface area, and the plant will give them in turn sugars. So it's good for both. Okay, that's one way. And then another mutualistic relationship is storing nitrogen-fixing bacteria in little root nodules. And they will provide nitrogen for the plant, and the plant will pay them back with sugars, because that's what a plant can get. Okay, and so then I also have root nodules, bacteria providing nitrogen. Okay, youngest bio buddy, tell them 2 things you've learned.
All right.
Yes.
No, what are the words like apple?
Oh, apoplast and symplast.
God, what's the
Apo. You know, like if somebody was talking in baby talk and they were going to say the word apple?
Yeah.
Apoplast.
It's apple.
I had a roommate in college, or a friend of a roommate, and she had a boyfriend who went to go, I brought you an apple. I can still picture it. Banana. And I'm like, how old are you? And I can remember being really annoyed by it. And from that, I've always remembered it.
Got it.
Apoplast. Okay. Did you tell them 2 things you've learned?
Yes.
Okay. So we focused on roots. Now we're focusing on stems. And you can have different kinds of stems. You can have herbaceous stems that are very green. And if they're green, they're doing what process? Photosynthesis. Or you can have woody stems that look like this. Now, if they're green like this, they'd be porous, and they'd have a way to get oxygen in order to do cellular respiration and CO2 to do photosynthesis. These don't. They have little openings, some there. They're called lenticels, little gaps and spaces. I don't know if I have it in this picture or not. No, I don't. That allows oxygen to get in there. So I'm just letting you know that. Here's a cross section, like of a tree. You don't have to learn all of these parts, so don't focus on that. But I want you to see right here. Do you remember how I talked to you about monocots and eudicots? And how the eudicots had in a stem, not in their root, but in their stem they have that ring, okay? They always make more xylem to the inside and more phloem to the outside. So as a plant grows, it always keeps generating xylem to the inside. The phloem might get broken off and you have to keep generating more phloem. When you cut down a tree, you're seeing everything to the inside. This wood, what we build houses with or furniture with, or in your shop class, that's all xylem. And you know how some wood is really soft, like you could scratch your initials in it, and other wood is a hardwood? It has to do with how thick the cell walls are. Okay, so heartwood is kind of old and clogged, and it's not being used for transport anymore, but sapwood still has sap in it, and the phloem is to the outside. This is this is called cork cambium, that's part that makes up the bark. So on your introduction to stems, number 3, adaptation, stiff stems will give support when there is no water, rising upward against gravity because more light is available. It is an adaptation that allows plants to move onto land. An adaptation that allows plants to move onto land. And there are many stem adaptations and modifications that have evolved in order to increase their ability to live in a certain environment or be successful in there. There are horizontal stems called runners, or stolons, strawberry plants. If their stem grows along the ground and you put soil on top of that stem, another whole strawberry plant will grow up out of it if there's soil that comes on top of it. Rhizomes, thickened rhizomes here in a potato, those are underground stems where you're storing those nutrients. Or here a corn, which kind of looks like an onion, is like an underground stem. Okay, and now I want to move on to leaves. Leaves have all different kinds of modifications. What would curling do? Why would they curl? Yeah, so if you're a woody stem, you would be supported by your thick bark on the outside, right? But if you're an herbaceous stem, right, you could only grow so tall and then you would what? Fall over, unless you're what? Hanging on to something. So by twirling around something, you can help attach yourself to it and get you at a higher elevation where you could have more sunlight. These are all things that you can use in essays to support the ideas of adaptations. Okay, so you can see this curling that takes place. Those are one type of leaf adaptation. Leaves here, you can see the veins. Would this be a monocot or a eudicot? Okay, and blue, talk about the differences here. We are so on target. You might be able to work on your lab today. Yes. Were you raising your hand?
Yeah, what's the Venetian called
For you to, it could be different. It could be palmate. I'll show you a whole bunch. There's a bunch of different names. Yeah. Okay. Introduction to leaves. Their purpose is for what? Photosynthesis. And then I just put highly variable. Okay. If you look at leaf structure here, what would be the adaptation of this space? Why do they have that space? Why aren't they just solid? Air, right? For what kind of exchange? Gas exchange, both oxygen and CO2. What they want you to do and understand, which is really good, is not memorize this is the structure, this is spongy mesophyll, this is palisade mesophyll in the leaf. That's not the critical part. The critical part is, why are they like this? What is the purpose behind that structure? Why is that an adaptive structure to have? By having that space that allows all of these cells to have access to gases in order so they can do photosynthesis and or cellular respiration. And they're not going to dry out. Their respiratory surface is not going to dry out because you have these openings right here. These are called guard cells that open up the, what do we call the openings? Stomata. And they will open and close based on the conditions in the environment and also hormonally and circadian rhythms. They will open and close under different conditions in order to allow gases in. But just as much as they can allow gases in and out, what's the risk you run by having your stomata open? Water loss, desiccation. What is that called when water evaporates out of a plant? Transpiration, which is not necessarily a bad thing, because water moves from the roots all the way up to the tops of the plant, the shoots, right? And what did we say? Water likes to hold hands. What is that? Yes, adhesion and cohesion due to what kind of bonding? Hydrogen bonding. So as the water evaporates, it's hanging on to another water molecule, which is hanging on to another water molecule, which is hanging on to another water molecule, right? So remember the analogy I gave you when we talked about the properties of water. If I'm a water molecule sitting right here and I start to evaporate out, what do I do with the whole column of water? What is that called? Yeah, cohesion when they're together. Adhesion is the support against the xylem walls, right? That will move water from the roots where you're getting it against gravity all the way to the top of the plant. Yes? So transpiration's good because by evaporating, it's pulling that water up so that the whole plant gets what they need. So you have to balance the need for water up through the plant with desiccation and drying out. You can't be permanently open, so that's why they will open and close at different times. Also, this cools a plant, because if it's a hot, hot day, the water in the ground is going to be much cooler, and you're going to pull that cold water up through the plant, and it acts like an air conditioning system. All right? So that is leaf structure. And here you can see different kinds of leaves. Simple leaf is obviously one blade. You can see this is compound leaf but it's, do you see what it is here? Palmately compound, like the palm. It's palmately compound, but it has pinnate venation in it. So those kind of words get thrown around here. This is pinnately compound and it's pinnate venation inside. So talking when it's compound is when there's more than one blade and it can be arranged in different ways. And I gave you some different pictures. Again, these are all just different adaptations plants have in order to acquire the light they need in order to do what process? Photosynthesis. Good. All right, and there's some other ones here, and then there's some other ones here. Okay, so leaf modifications. Cactus. Cactus have these really thickened leaves with spines, and why do they do that? Pardon? Get as much water as they can, and then the spines would be to what? If they're holding water in there, think about it. They've got water, source of water. They don't want animals to eat them. What kind of animals would eat them? Starts with an H.
Hyenas.
Herbivores. Go, Jeremy. Way to be varsity bio. Okay, so that is one adaptation. Here's a cucumber. You can see it's kind of curling around. Here's a Venus flytrap. Venus flytrap. What is it trapping? Flies. Why? Because Venus flytraps and things like pitcher plants tend to live in soils. They have an adaptation to overcome nitrogen deficient soils. Plants need nitrogen. And they're like, I want to live here. There's less competition here, but there's no nitrogen in this soil. So they have an adaptation by which they get their source of nitrogen by trapping insects and then digesting the insects for their nitrogen since they can't get it from their soil. Okay, another adaptation to overcome and exploit a different environment. Oh, and there's some close up. And here is a question. Oh, and I told you didn't even know this, but do you know this? Plus it is in your notes. Good, most of you got that? Good. Fish are friends, not food. She wouldn't stop shaking the bag. I shall call him Squishy. Yeah, you're not going to see those root hairs until you're in an area of maturation, because if you're elongating and you have a root hair on you, as you're elongating, what can happen to that root hair? It can get what? Ripped off, stripped. Yeah, exactly. Okay, we don't need to worry about that question. What's on the outside of the leaves to protect them from water loss? Waxy cuticle. Good. Five, 4, 3, 2. Mesophyll. Okay. How would you differentiate between A and C? Are they not the same thing? Yeah, epidermal, epidermis, that would be on top. So I don't know how you would pick. I'm just giving you strategies if you, when you're taking your AP exam, there would be no difference there. What do guard cells do? We talked about this already. Opening and closing. So is their job primarily photosynthesis? No. So even if you didn't know, then you could troubleshoot and get B for mesophyll. Tell your bio buddy, what were you thinking? Do you see it now? You're looking for parenchyma? Yeah. It's okay, you don't need to know that anymore. It's okay. Yay! The end! Okay. Good job, you guys. Super proud of you. And let's move on. Let's see, what am I doing?